You ever wonder how a plant, with just sunlight and water, can cook up something as complex as a cannabinoid? For decades, scientists had a rough idea, but the exact recipe, that was still a mystery. Well, today we're going to dive into the brilliant study that finally cracked the code.
Right, so how exactly does it work? The big question is about the precise molecular assembly line the cannabis plant uses to build molecules like THCA. That's the natural stuff in the plant that becomes THC when you heat it up. It's this fundamental puzzle, and, solving it took some serious molecular detective work. So to really get the mystery here, we first need to meet our main characters, the cannabinoids themselves. And these aren't simple things. They're really sophisticated molecules that get built inside these specialized little chemical factories in the plant.
What's so fascinating is the two-sided nature of these molecules. On one hand, you've got cannabinoids like THC, which are, of course, famous for their psychoactive effects. But on the other hand, there's this huge, and I mean rapidly growing scientific interest in their medicinal potential for everything from pain relief to controlling nausea to helping with tremors in MS patients, and even as a potential therapy for arthritis. They're complex molecules that play really complex roles.
Okay, so here's the crucial part. A cannabinoid isn't built from just one piece. It's a hybrid. Scientists call it a terpenolic compound, which sounds complicated, but the idea is actually pretty simple. Think of it like a car, built by welding the front half of one model to the back half of another. One part is a terpenoid, and the other is phenolic. For years, the big question mark was hovering over that first part. Where in the world does the terpenoid half come from?
And this brings us right to the heart of our detective story. In the world of plant chemistry, there were two main suspects, two possible molecular pathways that could be responsible for building that terpenoid piece of the puzzle. For the longest time, there was really only one suspect, the mevalonate pathway. This was the established textbook explanation for how plants built all their terpenoids. It was the reigning theory, the prime suspect, based on decades of solid research. But then a new clue surfaced. Scientists found a second, totally different assembly line in plants called the DOXP pathway. So now suddenly we have two competing theories. You've got the old guard, the mevalonate pathway, which operates in the main part of the cell, the cytoplasm. And then you have the newcomer, the DOXP pathway, working inside these specialized little compartments called plastids, the same place photosynthesis happens. The mission was clear. Figure out which one of these cannabis sativa was actually using.
Now, to solve this case, scientists couldn't just, peek inside the plant. They had to design this incredibly clever experiment to basically trick the plant into leaving behind a trail of evidence, a set of molecular fingerprints that would point directly to the true culprit.
The plan they came up with was just beautiful, a simple four-step process. First, they got some young, healthy cannabis sprouts. Second, they fed them a special diet. Glucose, but with some of the normal carbon atoms swapped out for a slightly heavier, traceable version, called carbon-13. Third, they just let the plants do their thing for 10 days, using this tagged glucose to build new cannabinoids. And finally, they harvested the plants and pulled out the finished products for some deep forensic analysis. This whole technique of using carbon-13 is called isotopic labeling, and it is the absolute heart of their strategy. It's basically like dumping a special, harmless colored dye into a city's water supply. If you then check the faucets all over town, the color of the water tells you exactly which pipes it flowed through. In this case, the carbon-13 atoms are the dye, and the plant's biochemical pathways are the pipes. All right, so the experiment is done. Now it's time for the moment of truth. Analyze the cannabinoids and see where those tagged carbon atoms, or colored dye, actually ended up, because where they landed would tell the whole story.
The key forensic tool for this job is something called NMR spectroscopy. You can think of it as a super advanced scanner that can pinpoint the exact location of every single carbon-13 tag inside the final, fully built cannabinoid molecule. It's like a high-tech fingerprint reader for molecules.
Now here's the cool part. Before they even looked at the results, the scientists had a really clear prediction. See, each pathway has its own unique way of building things, so it leaves a unique fingerprint. They predicted that if the newer DOXP pathway was the culprit, those special carbon tags would only show up in 4 very specific spots on the molecule. It's like predicting that the dyed water will only come out of 4 specific faucets in the entire city. And when the results came in, it was a slam dunk. The scan revealed that the carbon tags had landed in the exact 4 spots that the DOXP pathway model predicted.
It wasn't just close, it was a perfect, undeniable match. The molecular fingerprint left behind by the plant was identical to the one belonging to their #1 suspect. And the evidence was just overwhelming. The analysis showed that the terpenoid part of the cannabinoids was built, and get this, entirely or predominantly, we're talking more than 98%, through the DOXP pathway. The old theory was officially out. Case closed.
But wait, there's more. The study did more than just solve the main mystery. By tracing all those carbon atoms, the scientists also confirmed where the other half of the cannabinoid molecule came from, that phenolic part. As it turns out, building a cannabinoid is a team effort between two totally different factories in the plant. So here it is, the whole shebang, the complete confirmed blueprint. On one side, you have the DOXP pathway making a starting block called geranyl diphosphate. At the same time, a totally different assembly line, the polyketide pathway, makes another key piece called olivetolic acid. Then the plant takes these two big pieces and fuses them together. Two separate lines, one final product. Pretty incredible. And this discovery, it wasn't just some minor correction in a textbook. It was a fundamental shift in how we understand one of nature's most complex and talked about chemical factories. If you look back, you can really see how science builds on itself over time. Back in the 70s, experiments hinted at the old mevalonate pathway, but the evidence was always kind of weak. Then in the 90s, the discovery of this whole new DOXP pathway opened up an exciting new possibility. And finally, in 2001, this incredibly elegant experiment provided the definitive proof, settling a scientific debate that had been going on for decades. So why does all this matter? What's the bottom line? Well, for one, it solved a huge, long-standing puzzle. We now know for sure that cannabinoids are built in the plant's plastids using this DOXP pathway, and we've confirmed the other half of the recipe too. But most importantly, this gives scientists the foundational blueprint. And once you know exactly how something is made, you can start thinking about how to make it better, more efficiently, or maybe even create entirely new versions. And that leaves us with this one final fascinating thought. For the very first time, we have the complete and correct instruction manual for how cannabis builds its most famous molecules. So the question is no longer how, it's what's next. What new doors does this knowledge open for medicine, for agriculture, and for science itself? That, right there, is the next chapter just waiting to be written.